Baik adik-adik dan para peserta, seperti disampaikan tadi bahwa Dr. Pung adalah senior lecturer di Institute of Ocean and Earth Science atau IOS Universitas Malaya, Malaysia. Dan penelitian beliau telah memiliki banyak penelitian di bidang taksonomi molekuler dan filogenetics. Dan penelitian beliau sampai saat ini menggunakan omics tool, di mana mereka terdapat ada penelitian mengenai ekspresi metabolit dan gen, kemudian genetic diversity and population untuk ikan dan seaweed, kemudian fisiologi alga, kemudian bagaimana biochemical stress response dan adaptasi dari alga untuk menghadapi perubahan iklim dan sebagainya. Saat ini beliau menjabat, beliau pernah menjabat juga sebagai ahli jawatan kuasa keselamatan dan kesehatan pekerjaan atau JKKP. Kemudian beliau juga pernah menjadi chairperson of Committee for Procurement of Supplies Technical Evaluation sampai tahun 2021. Kemudian beliau adalah Committee Member for Procurement of Supplies Price and Technical Evaluation pada tahun 2020. Baik adik-adik, sebagaimana diketahui pada hari ini kita akan mendapatkan kuliah mengenai DNA barcoding, dan untuk itu saya langsung akan memberikan kesempatan kepada Dr. Pung untuk menyampaikan materinya. Dr. Pung, I'm sorry if I mispronounce your name. Should I call you Dr. Pung or Dr. Wan? Is that okay?
Dr. Pung. You're right, actually. Yes.
Okay. Okay, Dr. Pung.
Okay, so
you may begin your lecture today.
Okay, thank you, Dr. Novita. Right, so good afternoon to everyone here. So this is the second part of my lecture. We had the first part on last Monday. Okay, so let me share my screen. Okay, so I think everyone can see this right now.
Okay. Yes, we can see it clearly.
Okay, just a second. Right. Okay, so last week we did the first 5 topics. Okay, we stopped at how we search the gene bank or NCBI using the sequence that you have generated. Okay, so that is for rapid identification. If you want, then there's a few more steps to do. Okay, which will lead us to the generation of a phylogenetic tree. Okay. Next, right. Just give me 1 second. Okay. All right. So last week, how do I get rid of this? Okay. All right. So last week we already looked at this. Okay. I told you that after you blast your sequence against NCBI, we look at the percentage identity. Okay, so if you get something that is more than 99%, then you can be quite confident to say that, okay, this sample is actually the species listed here. So it's a Kappa-ficus malaceus. All right. But if you have another sample and after you blast it, you get the percentage identity of like 95%, 94%, okay, and so on. I wouldn't be very confident to say that this is actually Kappa-ficus malaceus. Okay, anything that is less than 99%, I don't think it will be a conclusive identification. Okay. So what we do in this case is we need to generate a phylogenetic tree. And if you ask me, scenario will happen. You actually went to the field and you collect a new species. Okay, so it has not been described before, or it has been described before in the past. Okay, you remember that many of the species that is available at this point have been described hundreds of years ago, and some of them have not been reported again. Okay, so so this might happen. Okay, so I was telling you just now, it could be a new species, or it could be something that is described before but have not been sequenced, or it could be something that has been sequenced but have not been deposited in gene bank. So that's why when you use your sequence to blast against what is available in the reference, you have very low percentage of similarity. Okay, this might happen. So in this case, you need to run a phylogenetic tree. Okay, so this is how a phylogenetic tree looks like. Basically, it's a tree. This is the backbone of the tree, and then the fonts in blues are the statistical support. Okay, so of course, the higher the support, the more reliable the grouping is. Basically, we want to see the grouping. So like this one over here, um, this actually corresponds to a single species. And then you have another group here, KS1, KS2. So this one is another species. And then within this species, there are two strains, okay? And then this is another species. And then this is the Eucuma lenticulatum, or what we commonly call spinosum. Okay. So before you can run your phylogenetic tree, you need to have a sequence alignment. Okay, in this sequence alignment, you have your own sequence and also some reference sequences. Okay, so that you have all these things in your tree, because you only have one sequence. And what about the rest? The rest comes from NCBI or gene bank. So you need to use your own sequence to compare with other sequences from gene bank to tell us where does our sequence group in which clade. Okay. So in order to search for reference sequences, I would advise my students to search from literature. Okay, you read your publications to find out within that genus how many species are available. And another option that is very useful is to search for databases. So if you work with fish, then probably you have heard about FishBase. So it's a catalog of telling you what are the species available, where are they distributed, and so on and so forth. Okay, so for algae, okay, because seaweed is a macroalgae, so I look for algae base. Okay, so what I do is I would search for Carpophycus. Okay, I would type in Carpophycus over here, and then I will search for the available species. Okay, so it's listed here. For the genus Carpophycus, there's only 6 species that has been described, but actually Carpophycus cottoni is a synonym. Okay, it has been already transferred to a different genus. There's only 5 Carpophycus. Okay. All right. So apart from searching for databases, you are also recommended to search for what sequences are available in NCBI. Okay, so let me share the screen of. Okay, hold on. Where is my? Okay, so you go to NCBI, click nucleotide. Okay, so if you know what is the genus, then you type the genus. Okay. Okay, so if you can see here, there's over 2,000 entries for copper ficus. So if you're going to see one by one, it's going to take time, right? So what I would suggest is, if you know what your genus is and you already know which molecular marker you want to use. So in our case, we use COX-1. If you type copper ficus COX-1 and then you search it, then it will narrow down to 295 sequences. Okay, these are the reference sequences that you can use, but it doesn't mean you have to use all 295. I will tell you how to filter among this 295 because it's of them are this. Now go back to my slides. Right. Okay. Okay. Okay, so I was showing you this just now. So if you're not sure what the genus is, you can search for the family. So if I type Soleriaceae, okay, if you look here under nucleotide, there's 12,272 sequences. There's too many, right? So it's not possible for us to look at it one by one. So what happens if I type Soleriaceae COX-1? It will narrow it down to 307. Okay, so you can either use the family together with the genetic marker, or you can use the genus and the genetic marker. So it will narrow it down further to 294. Okay, so another factor that you need to consider when you select your reference sequence is the length of the reference sequence. Okay, berapa panjang. So if you remember last week, I told you that what I sequenced was around 1,400 base pair. My COX-1 OMD 13 sequence was 1,004. So if you look here, this copper ficus inermis is only 700 over base pair. So if you want to use this, your reference own sequence. So what happens is you need to cut your half of your own sequence, okay, so that all of the reference sequence and your own sequence will be the same length. Okay, obviously yours is longer, so you have to cut yours. If the reference sequence is longer and yours is shorter, then you cut the reference sequence. Okay, so if in this case, the reference sequence is shorter, then you need to sacrifice your own sequence. And I usually avoid sacrificing my own sequence because the longer the sequence is, the more variation for you to run the phylogenetic tree. Okay, so one thing is you need to consider the length of the sequence. But actually for Kappa ficus inermis, what is available in GeneBank is only 700 base pairs. So it's up to you whether you want to include this species or not. Okay. All right. So my reference sequences. Okay. Bear in mind that anyone, okay, can submit sequences to GeneBank. I will actually show you, if we have time later today, how to submit sequences to GeneBank. Okay, so anyone can actually do it, but GeneBank only verifies, okay, they only check whether the sequence is technically correct or not. Okay, so if it's a COX-1 sequence, means if it's let's say a spacer gene, it's not a gene, if it's a spacer region, okay, then it's non-protein coding. So if it's a protein coding sequence, then it can be translated into amino acid, right? If you remember your biology lessons. Okay, so what GeneBank does when you submit your sequence is they check whether this is technically correct. Okay, whether you get your 5' and your 3' orientation correct or not, whether it can be translated into amino acid or not. Okay, so this means that I can say actually quite a number of sequences in gene bank are actually wrong. Okay, so they actually do not reflect the actual species. So how do I select which one to use for my phylogenetic tree analysis? I would suggest that you prioritize sequence of type species from type locality or from the verified distributional range. Okay, what does it mean? Okay, in your species, your scientific name, you have genus and species. Okay, so under the genus Capophycus, let's say, for example, Capophycus alvarezi is the type species. Type species means it's the first species described under that genus. So I would, if there's, let's say if just now I search and then what come out is Capophycus alvarezi, inermis, and maybe striatus. So I would prioritize Capophycus. I am not sure. And it should be from the type locality. Type locality means this sequence is generated from a sample that is collected from the first place where this species is described. So Capophycus alvarezi is actually first described from Malaysia. Okay, so I would, if there's a few Capophycus alvarezi sequence, one from Malaysia, one from Philippines, one from Mexico, then I would use the one from Malaysia because it's more likely to be correct. Okay, and then the verified distributional range means, okay, later I will show you one example of a sequence of Capophycus alvarezi, which is collected from Costa Rica. So Costa Rica is a country in Central America. So if you are actually familiar with Capophycus, okay, there's probably you should, because Capophycus is heavily farmed in Indonesia, right? Okay, so Capophycus, Capophycus is naturally available in Southeast Asian countries like Malaysia, Indonesia, and the Philippines. So if there's a sequence from Costa Rica on Capophycus alvarezi, it means that most likely this Capophycus alvarezi from Costa Rica is introduced from Southeast Asia into Central America. So I would try to avoid sequences that come from regions that this species does not occur naturally. Okay, I hope you get what I mean. And the fourth point that I want to say is try to, unless you're sure that, okay, then it will be less likely to be misidentified. Okay, what do I mean by unpublished sequence? So unpublished sequence means someone actually deposited this sequence in GeneBank, but this sequence has not been included in a publication. Okay. All right. So the last thing that I want to remind you is that GeneBank, they don't really have the manpower to keep on updating the species name in the sequence that you submit. Okay. If new publications come out, probably they will just update the publication status from unpublished to published, but they don't really update the species name. Usually, we as the sequence depositor, we will have to write in to GeneBank, email them, and tell them that, okay, my publication is out. I want to update my species name of the sequence that I have deposited to GeneBank. Okay, so you need to do this manually. So most of the time, the species name that you see on the sequences deposited in GeneBank is not updated. So what you need to do is you always refer to the latest publications. So the latest publications will show, by right, it will show the correct or most updated species name. Okay. All right. If you have any questions, please type into the chat. I don't see any questions at the moment, so I will continue. Okay, so let me show you an example of unpublished sequence. So this is So let me zoom in. So it's if you have seen a gene bank entry before, okay, you will see something like this. So this is the species name. This is the DNA marker that was used to generate this sequence. Okay. Okay, do you know why this is so long? It's because this sequence is actually the COX2, COX3, between the two genes, between COX2 and COX3, the spacer region. So it's called intergenic spacer. So this intergenic spacer, it is flanked by part of the COX2 and then part of the COX3. So that's why it says here, COX2 gene partial coding sequence, and then this COX2, COX3 intergenic spacer, complete, all right? This is complete. And then the last part is the COX3, also partial. So it's a little bit of COX3, a little bit of COX2, and the region between these two genes, okay? So what you can see from the gene bank entry is what is important is this is the accession number. Okay, it's the same here. So the 0.1 here means the version the version that they update. So usually we just take we don't take the 0.1, 0.2 at the back. You just take KR 063738. So the length of this sequence is 259 base pair. Okay. What else you need to know is, okay, look here. It says here it's unpublished. Okay. Usually when we submit the time when you submit this sequence to GeneBank is usually unpublished. Is accepted by publish. Okay. So this is the one that I was telling you. Okay. Okay, wait. All right. Oh gosh, how to get rid of this? Okay. So it says here this gene is the sequence is from the organelle mitochondrion, okay, because COX1, COX2, COX3 is a mitochondrion gene. So the molecular type is genomic DNA, and then the sample is from Costa Rica. So this is Central America. So remember this because later when you want to submit your own gene, your own sequence, you can also include this. Okay, you can say that the sequence is from Indonesia, okay, or you can say it's from Lombok, Indonesia. If you want to be very specific, you can include the collection date if you have it, and then who is the collector. Okay, so all this you can add when you want to submit and deposit your sequence. Okay. What else I want to tell you? Okay, so if it is a protein coding gene, okay, COX1 is a protein coding gene, then you can translate it to amino acid. Okay, so of the 259 base pair sequence, the first 82 is actually part of COX2. So COX2 is a protein coding, that's why it has amino acid. Between 83, no, between, yes, 83 until 222 is the intergenic space of protein coding gene. That's why it doesn't. And then the last part is the 223 to 259. This is the part of the COX3 gene. It's also a protein coding gene. That's why you can see the translated amino acid. And this is the DNA sequence. All right. Now let's look at an example of a published sequence. Okay, so I give you this example over here. This is a COX1, excuse me, COX1 sequence of Kappa ficus striatus actually. So when they deposit it, they put it striatum, but actually the updated name is striatus. So this shows what I mean by the name in GeneBank is always, it's most of the time not updated. Okay, so it should be striatus, not striatum. Okay, so you see here, this is the publication details. So this shows that this sequence is already published in which journal and then the title of the publication. All right. So if I were you and I have an option between a published and a non-published sequence, I would always go for the published sequence because this paper has been peer reviewed. Okay, unless you don't have any option, then you can use the unpublished sequence, but on condition that you check that the sequence is generated from authors that are expert in that field, okay? And also that it's within the native, or it's within the usual distribution. Now, I remember, let's have a look at the, okay. So I gave you one assignment, and you were supposed to do part 1 and part 2 last week. I have not received any emails from the organizer or from any of the participants. I hope you have done the assignments. Okay. Yeah. So part 3 is what I want you to do today. So I want you to search for the FASTA format sequences of these session numbers. Okay, because we want to use it for no. Yeah, some of them will be the reference sequence that we will use to generate our phylogenetic tree later. Okay. Let me show you how to do this. Okay, I'll show you for this sequence. Okay, KC9. So you just copy this, right? Okay. Go to NCBI, paste it. Okay, so it will come out the page for KC 905230. So what I told you is I want the FASTA. So this is not the non-FASTA. Non-FASTA is giving you all the information that you need. But if I just want the DNA sequence, I will click FASTA. Okay, so this is the FASTA format. Okay, FASTA format has this arrow over here, the session number and the name of the sequence, followed by the nucleotide. Need to copy, paste it onto a blank notepad. Okay, so this is the assignment that I want you to do, right? Find all the session numbers. Oh no, wait. The assignment is, oh, okay. I want to know the species name and also the locality. Sorry, I confused this with the later part. Okay, so if we go back to NCBI, okay, go back to gene bank. Okay, click gene bank. So this is the species name. So you're supposed to write Capophycus alvarezi, and the locality means where is this from? You can see here, it's from Lombok, Pengantap, Lombok, Indonesia. So you fill in the blanks with the answers here, okay? Pengantap, Lombok, Indonesia. And then the species name is Capophycus alvarezi. So please do this after the lecture. Not now because I need to continue with the lecture, okay? So let's go back to my slides. Hang on. Oh, where's my slide? Okay, here. Okay. Now, you have your sequence from last week. You have, now we need to do so as the name suggests, of more than one sequence, okay? Multiple sequence. So we will align your own sequence together with the reference sequences that we find in GeneBank to form an alignment, and then this alignment will guide us to run the phylogenetic tree. Okay, what is important here that I want to say is that in the alignment, okay, each of the column, it shows the same position in each sequence. So let me show you an alignment later so that you can understand better. Okay, and when you do your alignment, don't forget, apart from the reference sequence, we need an outgroup. Okay, what is an outgroup? An outgroup is a sequence that is not closely related to your own sequence and also the reference sequence. So just now, the OMD 13, okay, the one from last week, when you blast it, is Kappa ficus malationis, right? So if I want to prove whether it is really Kappa ficus malationis, I will search my gene bank, okay? I will search for sequences that are available for all species of Kappa ficus. So it's Kappa ficus, let me see. Okay, so there's alvarezi, there's inermis, malationis, procrustianus, striatus. But for information, procrustianus is not available in gene bank. No one has sequenced this before. So I will only include alvarezi for my comparison. So, right. So if someone asks why I need to do multiple sequence alignment, okay, because once you have your alignment, it will allow you to see which one is the conserved region, okay, because you have many sequences that is aligned. When you say align, means you susun, right? So when you arrange this, each of the column is showing the same position, and then each of the row is different sequence. Okay, so when you have your alignment, you will see which part is most conserved. Most conserved means they are all the same. So let's say you have 5 sequence, then all 5 sequence at that position shows A, or T, 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 T. Okay, so that is mean by conserve. So it's either conserve or variable. Variable means all of them are different, or some of them are different across the same column. Okay, so why you want to search for conserved regions? It's very useful when you want to design your primers. Okay, so when you design your primers for a sample that is unknown, but let's say I know it should be Kappa ficus, but I don't know what species it is, then I would do an alignment of DNA sequence from this different Kappa ficus species, and then I look for the conserved region, use this to design my primer. So when I use this primer for PCR, then I can amplify across the different species. It's not just specific for one species, but it can be used for different species of Kappa ficus. So it's more useful. Other includes you want to infer phylogenetic trees, okay, like what we're going to do later. So it also tells you an estimation of the evolutionary distance, whether they are closely related or they are very distantly related. So if they are grouped together, just now you saw the phylogenetic tree, right? So if they are grouped together in the same clade, means they are more closely related in terms of evolution. If they are more separated in the tree, one is at the top, one is at the bottom, means they are evolutionary more distant. Okay, so those at the bottom we call the basal part is what is available or should I say exists longer than those that are placed at the top of the tree. Those are more uh, those are how do I say, they exist much more recent compared to those at the bottom of the tree. Okay, so it also tells you which are the variable sites. Of course, if you see conserve, then you can also see variable sites. And sometimes you can also use it to uncover changes in gene structure. Okay, is there any questions? No? Okay, good. Let's continue. Okay, this slide is just to give you an introduction of the software cluster, which I'm going to use later. I believe you have downloaded this. So it has many generations, and the generation we're using is cluster X2. Okay. This together, cluster software, right? So I want you to open your BioEdit software, and then we are going to bring in the OMD13 COX-1 sequence that you have trimmed the front and the back last week. If you did my assignment, then you should have this, okay? So we are going to bring the COX-1 sequence of OMD13 into BioEdit, and then we are going to select some reference sequences to be aligned with our own sequences. So I have actually specified which ones I want, okay, to make your life easier. These are the ones that I want you to search in GenBank and then click FASTA to copy the FASTA sequences. All right, so let's do this together. Okay, so last week I have my OMD13. Okay, so this is the sequence that I have from last week. Okay, I told you that I already trimmed off the front part and the back part where it has the primer sequence. I don't want that. I don't want the noisy sequence. And I have also gave it the name of OMD13 COX-1. So what I do now is I'm going to copy this, right? And then open my BioEdit. Okay, so let me close this. So open your BioEdit software or your program. Okay, click New Alignment. All right. Oh, you're not. Are you not seeing this? Hold on. How come it's not showing? Oh. Okay, so you see the BioEdit right now, right? So just now what I did is I opened my BioEdit and then I click New Alignment, right? Okay, so just now I already copy paste my OMD13. So what I do now is I click import from clipboard. So it will come in here. Okay, so I already copied it here. You cannot click copy there and then paste here. I don't think it will work. Yeah, it doesn't work. So you have to use import from clipboard. Okay. Sorry, just give me 1 minute. I need to switch off my aircon for a while. Okay. So now you have your own sequence. What we need next is the reference sequence and also the outgroup sequence. So okay, so I want you to search for these sequences. We start with KC 905230. Okay, actually this is, I already searched. So, okay, I don't know why my slide is not moving. I think it's okay, oh, I didn't share my screen. Okay, so go to NCBI. All right, then search for this. Okay. Okay, search for this, and then I want the FASTA format. Okay, copy this, and then bring it to BioEdit, import from clipboard. Okay, so it brings in the reference sequence. So we do this for the rest of it. Okay, so let's do another one. KC 905238. Go to GenBank, paste the accession number. Okay, paste the session number, search, click FASTA. Okay, Excuse me. Then go back to our bioedit. Okay, so import from clipboard. Okay, all right. So you can carry on with this later. Okay, I will share the slides. You can do this. What I want to tell you next is okay, I need to find the outgroup. So outgroup is, let me see. So just now I was telling you that outgroup is something that is distantly related from the kappa ficus. So I will use Eucuma denticulatum as my outgroup. So let's see. I can't remember which one is denticulatum. Just give me one moment. Let me search. Outgroup. Okay, wait on. Okay, let me share my screen. Okay, this one. Is it, why is it not changing yet? Okay, I think you can see this, right? So this is what I have done earlier. I have copied everything from the slides, the session numbers that I've searched in GenBank. So I've copied them here, and this is my outgroup. Okay, KC905323. So and this is my own generated. Bring in the names. Okay, you will see something like this just now. Okay, so I want you to edit this name. Okay, you double click on the name because this one it is not going to be accepted when you use other programs to run the analysis later. We need to make this shorter. So first thing is I remove the 0.1 and don't use hyphen, use underscore. Okay, so all of this is kappa ficus, so I just put K to shorten it, right? Then I just leave the isolate number to differentiate between a species name. So the COX-1 gene, I can actually remove it because all of the sequence in my alignment is COX-1 gene. Okay, so I will make it shorter like this. I have my accession number, I have the species name, and also the isolate number. Okay, because if I just have, actually, it's not necessary. You can actually remove the L5 because each sequence has its own unique accession number, but I prefer to retain it lah for this case so that I know it is which one. So when you have shortened the name, click apply and close. Okay, so I want all the names to be tidy up like this. Okay, let's go back to my sample. So you see, I have already tidy up all the names, okay, like this. I retain the species name and also the isolate name, and most importantly, the accession numbers, okay? Now, what I do next is I'm going to copy all this sequence and then paste it onto a notepad so that I can use that file to run cluster, okay? So select all, right? Okay, then you have to click copy sequences to clipboard FASTA format. Okay, copy sequences to clipboard FASTA format. And then we open a notepad. Okay, so this is from before. I just removed this. Okay, you have opened a notepad, and then you just paste it. Excuse me. So I've pasted my sequences from Bioedit into a notepad. All right. I have to save it. So it's up to you what name you want to put. Let's say I put Cox1 for cluster. Okay, we're going to run cluster. It's really up to you as long as you know what file name it is. Okay, so save it. Oh, now we open cluster. Let me open my cluster first. Hold on. Cluster. Wait. Share screen cluster. Okay, so I have opened my cluster. Okay, I need to show you. Okay, this one. Okay, so you have your cluster. Now inside cluster, okay, I hope everyone is following me. Okay, no questions in the chat. All right, so click file, load sequence, and look for the file where you save just now that file. So just now I save it in OMD13. I put it as cost1 for cluster. Okay, so I open this file. Okay, so it looks the same as your bioedit, isn't it? So all the sequence, and then this is not arranged yet. Okay, so it's not aligned yet. Belum disusun lagi. Now I want to arrange it to align it properly so that all of them are showing the same position. So click alignment, output format options. Okay. Faster and also Nexus. Okay, click faster and Nexus. Okay. Okay, don't worry because this session is recorded, so I think the organizer will share the recording with you if you cannot catch, but try to do this together with me. So after I choose the output format options, now I want to do my complete alignment. So you click do complete alignment. Okay, so it will tell you that the tree will be saved to which folder. So it will be saved into the folder where you have your file just now, the notepad file just now. Okay, so click OK. You wait a while, it is running. Okay, so it's already finished running. So as you can see here, it cluster has helped me to align so that each of them is showing the same position. Excuse me. Okay, so you see, so your own sequence is longer than the rest. Okay, so later we're going to cut this part off and also this part. Okay, but we cannot cut inside cluster. It doesn't allow you to cut let's say, oh no, you cannot cut like this. You have to cut it in Bioedit. So what we do is we go back to Bioedit. Bio go to Bioedit. Okay. I'm not sure why it's not show. Okay. Oh, I haven't changed the screen yet. Okay. So now I want to open the file that is already run by cluster X. So click open. Okay, this folder. Go to the file where OMD13. Okay. You search all files, or else you cannot see. Oh no, it's not here. I think it's another file. Okay. So OMD13 just now, and then you will see this is the output files from. Okay, this file is the output file from cluster. Remember just now when we choose output format options, we choose FASTA, we choose Nexus. So cluster is it will by default give you ALN and DND file. Excuse me. So you have these 4 output files from cluster. If you're not sure, you can just look at the time. Okay, the time scale will tell you which one is the output file. So from out of to open in BioEdit. So this is how it looks like. All right. So if you want it to have color, then I just click this one, back colored view mode. Then you give me the color. Okay, so this is the same as in cluster X. And then you notice this extra cluster consensus sequence. Okay, this one, it has all these asterisks here, right? The column with asterisks is when all of them is the same. So this is what it means by conserve. So this is example of a conserve region. So where all of them is showing the same. So if I want to design my primer, I will design it somewhere here. Okay. One forward primer, and then maybe I want a reverse primer somewhere here. Okay, because it's conserved, I will design my primer using this part for the reverse primer so that I can get sequences from different species. I hope you can picture what I said just now about the consensus sequence. Okay, so I don't want this consensus to be used during my phylogenetic tree. So I remove this. I don't want this. Delete. Okay. What I want is all this. So in order to edit, I want to trim away this part. I don't want this part because I want every one of the sequences to be the same length. So what do I do is I click edit, okay? Then I highlight this part, click delete. And like the part that I don't want, click delete. Okay, so now all of them are the same length and the total length is 1407. Okay, my own sequence and the sequences from gene bank, they are all the same length. Okay, so now I copy them back into Notepad so that I can run the second round of cluster. I will tell you why I need to run 2 rounds of cluster in a short while. Okay, so copy sequences to clipboard FASTA format. When you want to bring something in, you click file import. When you want to copy something out from here, you click edit copy sequences to clipboard FASTA format. Then go to your notepad. Okay, open a clean notepad. Okay, I'm too lazy to open a notepad. I just use this one. So I paste it here. Okay, and then I give it a new file name. So just now we use Cox1 for cluster, right? So this is for cluster round 2. So I put round 2. Okay, it's anything you can put as long as you can differentiate which file is which file. Okay, so I put Cox1 for cluster round 2. Now, we go to cluster. Okay, so here, this is from the first run, right? Now we load sequences. This time, remember to load the cluster. Oh, okay, it's in another folder. Gosh. Oh, okay. Not here. Where did I put my cluster round 2? Where is it? Cluster. Where did I put my cluster round 2 sequence? Never mind, I will just save again. Give me a short while. Just my cluster round 2. Sorry, just give me a short while. Search for this. Where is it? Why is it missing? Out group with MD13. In the meantime, try to do what I asked you to do just now, if you can. Okay, wait. Where is it? Can't find it. Raw sequences Cox tree. Raw sequences. Raw sequences on D13. Okay, I think I know where it is. Oh gosh, why did I save it? Oh, okay. Right, I found it. Sorry about the. Okay, let's see. Cluster. Okay, so let me open the file again. Okay, Cox1 for cluster round 2. So it's already the same line here, but I need to run it another time because I need the output format for Nexus from this run. Okay, so I run it again. Make sure you do cluster file so that I can run the model selection for the phylogenetic tree. So you search OK. Okay, right. Just wait for a few seconds. It's quite fast. Okay, so it's already finished. Okay, so we close this, right? And then, okay, so this is the output format. Okay, what I need is the Nexus file, the NXS file. Okay, so now that we have our multiple sequence alignment already, I will show you how to run the tree. Okay. There's one question in the chat. Hold on, let me see what the question is. Oh, is there an attendance thing? I think this one, the organizer will have to answer you. Okay. Now, let me go back to my presentation first. Okay. Right, so I've already gone through this with you. Edit the name of the sequence because the software that we're using later are sensitive to symbols such as dot. It cannot have dot, don't have a hyphen. Okay, you use underscore. Try to shorten it as much as possible. Okay, you must have your, I would suggest you retain your session number because each sequence has a unique session number. Okay, so we proceed to multiple sequence alignment. So we have done this. Okay, I have showed. Okay. All right, so we will generate a phylogenetic tree next. Okay, what is a phylogenetic tree? Okay, it's also called a phylogeny. Okay, you see this tree over here. It's actually a diagram to show you the evolutionary pathway and the relationship between the organisms or between the taxa. Okay, so your phylogenetic tree may be built using morphology, okay, the body shape. Okay, maybe it's the length of the fin of the fish or the size of the bones or anything else that you can think of, the morphology features. Okay, in our case, most of the time you use DNA sequences. Okay, you can also use protein sequences to run the tree as well. Okay, and the latest update is some people even use RNA sequence to run phylogenetic trees. Okay, so it's not just DNA. Okay, so any DNA, RNA, or protein sequences can be used to run or draw a phylogenetic tree, but the DNA sequences are still the most common. Okay. All right. So generally, there are many types of phylogenetic trees based on what algorithm you want to use. Okay. There's maximum likelihood, which I'm going to show you today. There's also a Bayes tree, um, neighbor joining, or we call NJ. There's also maximum parsimony, or more is we usually use ML Bayes, and there's even something that is more newer, okay, ABGD and something else. But for today's class, I will show you maximum likelihood, how to generate a maximum likelihood tree. Okay. If you want to understand more about these different types of trees, then you can try to read from internet. Okay, I don't have time to cover that for today. I'm afraid. Okay. So how to obtain a maximum likelihood tree? Just now you have built an alignment. So the next step is we need to know which is the best model to infer the tree, okay, to draw the tree. Okay, after you have identified the model, we will run the tree. Okay, so now I will show you how to do this using this web page. Okay, IQ tree. Okay, let me. Okay, my browser. Where is my browser? Okay. Right. So, okay, I think you can see my browser now. So you see here, this is you can search for IQ tree and then it will take you to this web page. Okay, IQ tree. This is the web portal. There's also another one where you can download the software and run it in your laptop. But I prefer to use the online version. Okay, yeah. So you see here, there's tree inference, model selection, and analysis results, 3 tabs. So the first thing that we need to do is selection, because you need to data into this page. So alignment file, if you're not sure what you need to put here, it will tell you, you see browse, these are the formats that they accept. So it's Nexus, Cluster, MSF, show example. Okay, it will tell you can see this. So just now I have run the cluster, right? The second round of cluster. I want to get this Nexus format. Okay. You can actually use FASTA and cluster as well, but I prefer to use the Nexus. Okay. So now I click browse. browse and then go to the file where just now the cluster, you remember? Okay, Cox1 for cluster round 2. I take the Nexus file. Okay, open the Nexus file. Okay, so it's already here. This is our input file. Use example alignment. No, I don't have to click that. Sequence type, auto detect. It's actually DNA. Never mind, it will auto detect. If you want to click DNA, also can. Partition file, I will leave it empty. This one is when you have a few genes that we merge together, then we will tell it number 1 until position 100 is COX1, and then position 101 to 200 is another gene. Okay, but here for today, we're not using this, so just leave it blank. Selection criterion. Okay, there's 3 types. A Kaiki, corrected AIC, a Kaiki. For today I will use food. Basically these 3 types they are slightly different. And the difference is how do I say, the algorithm is a bit different. Okay, but usually they will give similar results. Okay, you can read more about this by searching for the manual, okay, of IQ3. Yeah, there's a tutorial here, or you can visit the IQ3 homepage if you want to read up more about this. It will tell you what is the difference. Okay, so for today, you just follow me. Tick corrected AIC. New model selection, no, just leave it empty. Okay, so now you have to type in your email address so that it will send you the results. Okay, so this one we just leave it, we don't have to fill it in. Okay, so just click, just input your file and then click corrected AIC and then submit the job. So it's submitting to the server. So once your result is out, then it will tell you, but actually it's quite fast. Okay, so let's see, run log. I think it's already finished. Or is it? Let me see. Full result. Yes, it's already finished running. Okay, so you should receive an email telling you that the result is out. Okay, so how it looks like, the results. Let me see, where is it? Okay. So these are the results that will be sent to you. Sorry, this is no, this is the ML3. This is okay. The model. Okay, it will send you these 3 files. Okay, the front part is the name of your file, of course. Okay, and then it has this IQ3 file and access and 3 file format. Okay, so let's see. Wait, let me just, I need to show you what is the difference. No, not this one. 3 file. Okay. AII model of substitution. Okay. All right. So we open the first file, the .iq3 file with Notepad. Okay. Okay, can you see this? Now, so the purpose we are running this is to know the which is the best model. So it will tell you here, best fit model according to AICC, corrected AIC is HKY plus F plus I, right? Okay, the rest, no, you don't have to look at the rest. Okay, so remember this is HKY plus F plus I. Now we go back to the browser. Okay, so now, okay, are you looking at the browser? All right. So now, after we do the model selection, now we want to infer the phylogenetic tree. Same thing, browse for the cluster file just now, the same one. Cox1 for cluster round 2, nexus file. Okay, auto detect. Okay, I'll just choose DNA partition file, optional, no, substitution file. Okay, it's a substitution model. So just now it already run for us. The best model is HKY plus F plus I. So you can actually, yeah. So if you click this question mark icon, it will tell you what is it lah. You can read about this. So I will choose HKY because just now it already tells me it's HKY plus F plus I. So in the plus F, I will show you how I get this empirical later. Okay, so the rest I just I don't click the rest that I'm not sure. Okay, so just now I told you that when you run your tree, you need to have the backbone of the tree and also the statistical support. So branch support analysis is to give you the support values, the statistical support. Okay, so you can either choose none, ultra fast, or standard. Okay, if you want to know more about this, you can read this. None is no bootstrap, standard is much slower, ultra fast is much faster. Okay, so I will choose ultra fast, and then I'm going to run 5,000. Okay, create this, no, maximum, nope. So this one, if you want to run the single branch test, you can. Some people wants to report this in their publication. Some people doesn't want. So it's up to you. If you want, you can choose as. Yes, I will choose no for this time. Right. So the rest I will leave it as default. Okay, let's see. This one, no. Okay, just leave it empty. Same thing, give your email address and then submit the job. Okay, it's very simple, isn't it? So wait for the result. Okay, so let me show you the result that I already run. Let's 13. No. Okay, so after you run, you should get an email and then it will ask you to download the results. So you just download and then you will see something like this. There's 4 files in here. The con tree, the IQ tree, the nexus file, and also the, this is the log file if I'm not mistaken. Yeah. So now what we want to do is we want to see our tree. Okay, so we will open this con tree in fig tree program. So is everyone catching? Hold on, let me see if there's anything in the chat. Oh no, nothing in the chat. Okay. So now let's open fig tree. I've also asked you to download this. Fig tree. Okay. Open fig tree. Okay. And then click open. Is it moving? Oh, okay. I need to share the screen first. Sorry. Okay, fig tree. All right. Okay, so we're now at fig tree. Click File, Open, and then go to the you downloaded the result. Okay, so just now my results, I put it here. Okay, this is the results. So I open the country format, the country file. Country means the consensus tree. Okay, so I open it. All right, so it will pop up something like this. Please select a name for this value. This is actually the support value. So you just leave it as label. Okay, click OK. So you can see your tree right now. Okay, it's not very nice at the moment because we need to modify it a bit. So the first thing I would modify is click trees, order the nodes. Okay, I usually choose decreasing, and then I will root my tree. Remember just now, when we do the alignment, I asked you to download the sequence of eucuma denticulatum. That is our outgroup. Okay, the function of the outgroup is to root the tree. So user selection, where is my eucuma denticulatum? It's this one. So I click this eucuma denticulatum and then click reroot to root my tree. Okay, root the tree means I want this to be at the bottom of my tree because it's the most distantly related. Okay, so I click reroot. Okay, you see now it is rooted at the bottom. Okay. All right. Next is what the root. So later I will cut this off so that it doesn't look so long. All right. Next is my support values. Okay. So you go to, this is the tip label. Tip label is the things, the names over here. Sorry, because my cursor cannot show you. So, okay, let me show you like this. If I unclick the tip labels, the names on the tree will be missing. So this one, don't move it, okay? But I click node labels, and then when the node labels, right, it has several options. So you click label. Remember just now when we opened fig tree, it popped out label, right? So this is the support value actually. So you click label. Now you will see the support value over here. So it's quite small right now. You can make it bigger. Oh, okay. This is, yeah. So it's a bit bigger right now. Let me make the tip label bigger. Okay, I think you can see better right now. Okay. All right. So you can see here the striatus is grouped together. The alvarezii is grouped together. Right. Okay. So if you want to show this tree in your publication, okay, your manuscript, your paper, we need to save this and then shift this into PowerPoint to edit it further before you include it as a figure in your paper that you're going to submit to journals. Okay, so what we do now is click File, Graphic. Okay, export graphic, and then click EMF file, Windows Enhanced Matter file, and then the name. Okay, put it in the same folder. Okay, so this is a consensus tree file, right? So consensus tree. Okay, just put whatever name you want as long as you know. Or okay, now I put, this is my ML tree, so I put ML tree. Okay, click OK. So it will have saved this file as a photo that you can open in PowerPoint. All right. Okay. Now let's look at, let's open a PowerPoint. Let me see. Give me a while. PowerPoint. Okay. PowerPoint. Okay, so I open a PowerPoint and then I click Insert Pictures. So you go to the file where you save your tree. Just where did, okay, MQ3. Oh, okay, where did I save it? Never mind. So it's actually the same as this consensus tree file. Okay, so let's open this. Okay, so this is the tree that you saved just now in Fig Tree. Okay. Click this, right? Because we want to modify this tree so that it looks more presentable to be included in my paper for submission to journal. So you click Ungroup. Okay, treat this as a photo. Click Ungroup. Okay, and then you can keep on clicking Ungroup until you see something like this. Okay, so now I want to make the font bigger. Okay, you can change it to, let's say I want Arial size. Okay. So now, if you want to edit, you can edit. You want to throw away the isolate name or whatever it is. Okay. But most importantly, what I want to show you today is the bootstrap support. So this one, you have to shift it here. Okay. So because it's nicer to look like this. So it's GUI 10. Relationship between them is close and the support value is 95. Yeah, I think you should be able to see. And then for this one, we shift it like this. Okay, so how do I know where to shift it? Actually, you can refer to your let me share my screen again. Hold on, let me search for okay, you can open just now this is the output file from IQ3, right? So you can open this IQ3 file. Use Notepad to open it. Okay, let me share screen. Okay. Share screen. Can you see it? Okay, I think you should be able to see this. So this is the IQ3 file. If you scroll to the bottom, okay, you see this is a tree, and then these are the support values. So it tells you this support value is between this one and this one. Malaysianist 141 and Cox OMD 13. And then the 77 actually refers to the relationship between these three. This tree. Actually move it to here. This tree. Okay, so we go back to our PowerPoint. So Malaysianist 141, 13, and Malaysianist P1 is 77. So I move it here. So you have to compare between your own tree from this PowerPoint and also the file from IQ3. Okay, and then anything that is below 50 and below, okay, or usually if it's 50 something, I would also take it away. Okay, I would remove it because the support value is too low. It's only 50% is reliable, so I usually remove the 50, 58. If it's 60 something, it's still not too bad. This is only 36, so I'm going to remove it as well. Okay, so this one, the 100 is actually here. It refers to L5 and Kappa Ficus alvarezi L5 and 40. Okay, so and then this one, I'm going to shorten it. If you see the publications, this is very, you don't have to be very long. Okay, so let's say, okay, I'm happy with this tree. Okay, this one, the 0.3 is the substitutions, the number of substitutions. Okay, so now that I'm already, no, actually you should, when you make the species name italic or do all this, the species name should be italic. And then this one, OMD 13, I can actually change it to Malaysianist. So Malaysianist, okay. So I don't have an accession number yet because I have not deposited into GeneBank. Later I will show you how to deposit. We don't have much time. Maybe I'll skip. Let me see later. Okay, so you see here, this 77 refers to Malaysianist. This three Malaysianist. 141, OMD 13, and uh this one, KC 905319. So we are quite sure that this is a Malaysianist, although last week I already showed you that when you blast it, it gives you 99% of Malaysianist, right? So yes, it is Malaysianist. Okay, so if you're satisfied with all this, remember to group it back. Okay, save it as a picture. Okay, save it as a picture, then later you can use it in your publication. Okay. Now, I need to go back to my slides. Okay, so we have done this. I have shown you how to edit PowerPoint. Okay. So everything that I've shown you just now, we use Chromas Pro, we use BioEdit, Cluster X, okay, to run our analysis and our trees. So sometimes if you want to use this software, personally, but yeah, I've heard that a lot of people use this as well. So you can actually explore this if you want. So this is what I was telling you just now. There are a lot of methods out there to delimit species, or what it means is to resolve different species. Okay, so if you want to know more about this, you can read this paper. I don't have time to cover this today. How to determine pairwise distance. Okay, we still have 10 minutes. I think I can do this briefly. So how to determine pairwise distance? When do you need to do this? Pairwise distance is when you want to describe a new species, or you want to tell people the genetic diversity between two different species. We use the pulp software. Okay, so we will use the cluster nexus output that you get from your cluster, the second round of cluster analysis. So that nexus format, that nexus file is very important. You use it for pulp, you use it for IQ3. Okay, so you open that file and then you paste the command that I have already shared with you through the organizer. There's one command for pairwise distance analysis. You open your cluster nexus output and then paste this command at the bottom. Okay, save the file and then run it in pulp. Okay, I'm going to show you how to do this right now. Let me see. Okay, Let me go back to my so this is my cluster file from the second round of cluster analysis. Okay, so I drag it to the bottom and then I paste this command. Okay, I already shared this with you. Okay, so you just copy paste into this cluster file, right? Save it. Okay, I save it as run p distance. Okay, now I open my pulp software. Open pulp. Pulp. Okay. Can you see it? All right. So you can see this right now. Open pulp and then select the file that you just saved. So just now I save it as run p distance. Oh, why is it always missing? Oh, okay. All files. Okay. Run p distance. Okay. Because I already run this earlier, so I just this, which is why it's asking again. Okay, so it will be the analysis will just take a few seconds. So it's done running. Okay, so let me show you how to interpret the results. PowerPoint. Okay. Okay, when you open your results from this pulp run, you will see something like this. Okay, this is the absolute distance file. So what it means like this is between OMD 13 and this MY216, it has one base pair different. Between INP1 and MY216, it has two base pair different. So this one, this column over here is the same as this side. So it's MY216 OMD 13 over here, INP1, GUI1, V15, INL5, MY40, GUI2. Okay. So it will tell you between the two sequence how many base pair is different. Okay. If you open the p distance file, which is also the output from pulp, okay, it will show you something like this also. This one is the probability. So if you want to change it to percentage, then you have to multiply by 100%. So example for this one, KSB, KC905299, and MY216KC905308, this column, this one and this one, the genetic diversity between these two is 6.32%. So if you want to report two sequence, percent, okay, this is useful when you want to do genetic diversity publication or you want to describe new species. Okay, last one that I want to cover for today is how do we deposit DNA sequences into GenBank. Okay, so let me share my screen from, let me see, where is my browser? Okay. Okay, let's go back to the home page. Okay. Okay, so you can see here, this is the home page of NCBI. So if you want to deposit GenBank sequence, you click submit. Okay, we want to submit DNA sequence. So you scroll down, So usually you would take this one because we are not submitting RNA or viruses. This is genomic DNA. Okay, so we suggest we take this one, submit genomic DNA. This one is for genomes, so we are not selecting this. Select submit. Okay, so for NCBI nowadays, they need us to do third-party login. So first you need to set up an account with NCBI first. Okay, so that you can log in. I have a login account, but I can't remember. Okay, never mind. I will show you my PowerPoint. It's easier. All right. Okay. So submit to GenBank, right? So we're going to use Bank It, right? This program, the tool. Okay, I already logged in. I can't remember my login details at the moment. So this is, I took this from when I've logged in. Okay, so you need to set up an account with NCBI and then you log in. Okay, so we click Start Bank It Submission. Okay. So they will come out a page here. There's many tabs, right? You have to go through all these tabs before you can submit your sequence. So the first page is to know about your own details. So you fill in your first name, your last name, your department, all those things. All right. Then we continue. Okay, continue. The second thing they ask you is reference. Okay, so this one means your publication, your paper that you're going to publish this sequence. So this, you have to fill in all the author's name from your paper or your manuscript if you have not published yet. Okay. This one, if it's not published yet, then you click unpublish. If it's already accepted, then you click impress. If it's already published, then you click publish and give the title. Okay. Sequencing technology, it's dideoxy sequencing. Okay, this is usually the case lah. Unless you sequence genome, then probably it's Illumina or IoTorrent, PacBio, those kind of things. Okay, are these sequences unassembled or assembled sequences? So usually I would go for, if I want to deposit one by one the sequences, then I click unassemble. If I want to submit an alignment, okay, of sequences, that I can search, I can click assemble sequences. So it depends on, you want to deposit single sequences or a batch of aligned sequences. Okay, continue. Then GenBank will ask you, when do you want to release this sequence? Is it immediately after they approve, or you want to give a release date? So usually we will put as long as possible, because we want our paper to be published before the sequence is released. But it's really up to you. If you want your sequence to be released immediately, then by all means go ahead. Okay. It doesn't mean you need to have your paper published before your sequence is released out. Okay. So molecular type, let's see. I think this is something about DNA. So topology is sequencing the chloroplast or mitochondrial genome, then it's circular. So it's linear. Are you submitting the complete sequence? Yes or no? Usually it's no if we sequence a molecular marker, okay, because you cannot get the full length sequence. So sequence data format, FASTA sequence, okay. If it's alignment, then you choose alignment. Then you paste your OMD13 sequence over here. Okay, or you want to upload the file also can. Okay, so organism, the name. Okay, type in the name. Submission category, original if it's sequenced by you. Okay, not from another person. Okay, so because this is a COX-1 sequence, right? So I will select mitochondrion. Okay, it's from a mitochondrial gene marker. So the source modifier is you can put the country name, you can put who is the collector, the collection date, all those things. You can add it here. Okay. Right, so you see country. Yeah, if you want to put the isolate name, then you choose cultivar. Okay, so there's different types of things that you can add in. You want to put the voucher name also can. The catalog number of the sample also can. All right. Okay. So add features by completing input forms. Okay. Basically this is to tell GenBank what is or if it's a spacer, then it's not a non-coding region. Okay. All right. So COX-1 is a coding region. That's why I put coding. So you type in the protein name. COX-1 is cytochrome oxidase, subunit 1. And then I will have to find the amino acid sequence. I'm going to show you how to find this amino acid sequence. You have to type it here. Okay. It's actually copy paste lah. You copy paste the amino acid sequence here. Okay. So this gives you the summary of the features. So it will tell you it's a coding sequence, and then this is the this is the amino acid sequence. So if you want to add other things, you can add other things. Okay, so this is how it's going to look like, right? When it's uploaded into GenBank. So you can check. This is review records of your set. So you can review first. If you're happy with it, then you submit. Click finish submission to submit. Okay. Okay, you see here, when you review submission, you can also say whether, okay, you see, you may update or revise your submissions at any time by sending an email to NCBI. Okay, so if you, after your paper is published, you want to update the names, then you can send an email to NCBI. And you have to put your corresponding email address so that NCBI can contact you if there's something wrong with your submission. Okay, so we're actually done. This is just to show protein sequences from the sequence, right? OMD30. You just go to this website, or you can Google any website that helps you to translate DNA into amino acids. Okay, it will translate for you. Okay, and then you have to choose the genetic codes. Why you need to choose the genetic code? Because I think you should know, right? Your protein, it has 3 frame shift, right? Codon 1, codon 2, codon 3. So you have to see one by one, the 3 among the 3. Check which one doesn't have stop codon in between. If it has a many stop codon, means this is the wrong frame. Okay, the stop, stop, stop all over the place. So this one is the frame 1 is the correct one. So I will take the amino acid sequence from frame 1. Okay. All right. So yeah, this is just to show you the codon and the amino acid. Okay, so I think that's the end of my lecture today. It's already 4:05. Is there any questions from the public, from the participants? No, I don't see any questions. Over to you, to the organizer.
Okay. Okay, thank you, Dr. Pung, for the insightful lecture today. Well, although excuse me, I've catch the influenza this morning. As I mentioned before, although I have never had a research that include the DNA barcoding, I think this tools is useful for research who wants to know the diversities or their objects. As I remember, I had molecular research at 2014
Mm-hmm.
that include the excuse me that include the I'm sorry.
It's okay.
Yes, I think there are a lot of people who catch the influenza disease. I don't know whether it's a common cold or something because a lot of people I know got the same I have molecular research on Capapicus alpha-rhizi, and we just and at that time I just isolated the DNA
Mm-hmm.
of the species and then do PCR analysis using COP1 and COP3 Spencer, if I'm
Mm-hmm.
not mistaken.
Yeah.
And the sample was just sent to sequence the DNA, and then the result was given to someone to do analyze the maybe they include this method or tool uh, to determine whether the species yes the exact species, but uh, sorry. Well, I'm sorry. Excuse me. Um, but I think your lecture has given us such light and knowledge about blasting or you know um, to determine our age or whatsoever. And I also think that your presentation and lectures today very useful for our students and participants. Um, Although there's maybe it's only one or two questions that delivered to you by participants. Maybe the participant could get the recording for these sessions by the organizer. And
Yes.
and or the organizer of uh, say thank you very much for your time,
You're
and
welcome.
uh, I am and for the postponing the meeting 15 minutes and maybe waste your time, I don't know. But thank you very much for your kindness.
Yes, thank you very much.
Yes,
Yeah, don't worry about it. I hope it's
yes,
useful for the participants as well. Yeah.
yes, and I think so. Dan adik-adik semua telah mendapatkan kuliah singkat tadi dari Dorfung. Tentu saja banyak sekali ilmu yang ada di
Mm.
yang adik dapatkan dari beliau, dan semoga ilmu tersebut bermanfaat buat kita semua. Once again, thank you very much, Apu, and we should close this meeting by reciting Hamdalah for the participants and Alhamdulillah. Okay. Thank you very much once again
Okay.
for this insightful lecture. And I hope someday we will meet or get another knowledge from you.
Yes. Okay. Sure. Sure. Yeah. Okay.
Okay.
All right. Thank you, everyone. Okay.
Baik,
All right.
Dr.
baik,
Pong,
akhir kata,
before we
Assalamualaikum
ending
warahmatullahi
this meeting,
wabarakatuh.
we are
Selamat sore.
from
Good
committee.
afternoon.
We would
Silakan
like
ke
to show
organizer.
our appreciate that we would like to share the certificate.
Okay. Oh, okay. Yeah. Thank you.
Thank you very much, Dr. Pong, on your lecture today.
Thank you. You're welcome. And then I just want to add that I welcome any one of the participants who would like to further their study at UM. Yeah, you're welcome. Just contact me if you're interested to do your PhD or your master's in Malaysia. Yeah. So thank you, everyone. Okay.
Thank you very much.
Thank you. Right. See you again. Bye-bye.
Bye. Thank you, Dr. Pong.
You're welcome.
Thank you, Dr. Pong.
Before we ending this meeting, would you like all of the participants to open cam? We would like to photo session. Please, all of the participants. Okay, ready? 1, 2, 3. Cheese. Okay, thank you very much for the time, Dr. Pong and
You're welcome.
Ibu Novita for the time. Thank you, terima kasih banyak.
Okay.
Oke, untuk semua peserta dapat meninggalkan Zoom meeting. Terima kasih.
Okay.